Hello guys and welcome back to Exonuclease Academy. So here I am with another interview type questions for practice based on the replication of DNA. So the question is, hypothesize why some DNA polymerases lack exonuclease activity without significantly contributing to the number of mismatches introduced during DNA replication. ये बहुत ही सिंपल है. सिंपल भाषा में ये पूछा जा रहा है कि जब हम एक DNA polymerase, like for example, we have talked about DNA polymerase 3, the core hollow enzyme. It has got the proofreading activity, but DNA polymerase 1, it does not have the whether or it lacks the exonuclease activity. ठीक है, तो ऐसा क्यों है कि एक के पास है प्रूफ रीडिंग एक्टिविटी एंड दूसरे के पास नहीं है. ठीक है, तो इसका क्या मतलब हुआ? सी कैसा होता है कि ऑलरेडी वी हैव मेंशन दैट देयर आर different types of DNA polymerase, right? Like for example, in the case of prokaryotes, we have got different types, like around 5 are there, and in the case of eukaryotes, around 13 types of DNA polymerases are there. ठीक है, तो हर एक DNA polymerase जो है वो स्पेशल प्रोसेस के लिए यूज होता है. फॉर एग्जाम्पल DNA रिपेयर. ठीक है, नाउ DNA रिपेयर में क्या होता है, दे डोंट नीड टू बी वेरी प्रोसेसिव and they do not have to carry out bulk amount of DNA synthesis, right? जैसे कि DNA pol 3 को करना पड़ता है. Whereas in the case of DNA repair, जहां पे भी मिस्टेक होता है, मिसमैच होता है, सिर्फ उसी को ही हटा के नए ऐड किए जाते हैं. ठीक है, सो इसलिए प्रूफ रीडिंग इतना इंपॉर्टेंट नहीं होता है फॉर सच काइंड ऑफ DNA polymerase, because they're going to add only small number of DNTPs, and hence the number of mismatches or the number of mistakes they are going to do is going to be negligible on the leading or the lagging strands of the DNA polymerase. ठीक है, so बहुत सिंपल सा सवाल था, but हाँ, just imagine you are in the interview and the panelists are asking such questions. So even though you know the answer, but doubt हो जाता है. So you have to give yourself, some time, take your time and just let them know कि हाँ sir मैं सोच रहा हूँ या सोच रही हूँ, or just give me some time, and then you can start saying, and then you can mention about like how exactly the DNA Pol 3 is working, how DNA Pol 1 is working, and then you can justify the question. ठीक है, sorry, justify your answer. So in this way you have to talk about. So just let me repeat once again. So we have you need to talk about DNA Pol 3, that it has to add lots of DNTPs. So the processivity in this case is going to be very high. So the chances of adding wrong nucleotide is high over here, and hence it has got the exonuclease activity. Whereas if you talk about DNA Pol 1, which is basically required for your DNA repair mechanism, right? So यहाँ पे क्या होता है, you just need to add small amount of DNTPs. So the chances of adding wrong DNTPs is going to be very, very less. And hence, there are certain DNA polymerase which lacks the exonuclease activity. ठीक है? So this is how you need to justify your answer. So guys, ये बहुत ही simple था, बहुत ही छोटा question था. Okay, so my effort was just to bring such questions in front of you so that it can make you think about it. And ये सारे topics जो हैं हमने cover किए हैं DNA replication में. So if you are missing any point, if you're not able to understand, just let me know through your comments. And please watch all the videos. There are around 17 lectures, which is completing the entire chapter 9 from the Watson. That is for your DNA replication. Okay, so thank you guys. If you like the video, please hit the like button and let me know through your comments. Thank you and take care.